Good afternoon, and welcome to Cara Therapeutics' fourth quarter and full year 2018 financial results conference call. All participants are now in listen-only mode. There will be a question and answer session at the end. Please be advised that this call is being recorded at Cara's request. I would now like to turn the call over to the Cara team. Please proceed.
Good afternoon. This is Jane Erheim with Stern Investor Relations, and welcome to Cara Therapeutics' fourth quarter and full year 2018 financial results and update conference call. The news release became available just after 4 p.m. today and can be found on our website at www.caratherapeutics.com. You may also listen to a live webcast and replay of today's call on the investor section of the website. Before we begin, let me remind you that statements made on today's call regarding matters that are not historical facts are forward-looking statements within the meaning of the Private Securities Litigation Reform Act of 1995. Examples of these forward-looking statements include statements concerning the expected timing of the completion and announcement of the results of our ongoing clinical trials, the expected timing and design of our planned clinical trials, future regulatory and development milestones for our product candidates, the potential for CR-845 to be a therapeutic option in multiple pruritus indications, the size of markets that are potentially addressable by our product candidates, and our expected cash reach. Because such statements are subject to risks and uncertainties, actual results may differ materially from those expressed or implied by such forward-looking statements. Risks are described more fully in Cara Therapeutics' filings with the Securities and Exchange Commission, including the Risk Factors section of the company's annual report on Form 10-K for the year ended December 31, 2018, and its other documents subsequently filed with or furnished to the Securities and Exchange Commission. Participating on this call are Dr. Derek Chalmers, Cara President and CEO, and Dr. Mani Mohindru, Chief Financial Officer and Chief Strategy Officer. I'll now turn the call over to Dr. Chalmers.
Thank you, Jane, and good afternoon, everybody, and thanks for joining us on the call today. 2018 was certainly a year of significant achievement for us here at Cara, including the advancement of our lead drug candidate, KORSUVA, across a range of clinical populations where pruritus is a significant unmet need, the signing of an important strategic licensing agreement with Vifor Fresenius, and the successful completion of a $92 million follow-on offering. So this clinical and corporate progress has certainly laid the groundwork for what we believe will be a transformative year ahead for the company. In 2018, we expanded our phase 3 program of KORSUVA Injection for chronic kidney disease-associated pruritus, or CKD-aP, in hemodialysis patients with the initiation of our 2 pivotal phase 3 trials designated CAM1 and CAM2. In January of this year, we announced we had completed enrollment of the CAM1 trial, and we remain on track to read out top-line data from this trial in the second quarter of this year, as well as top-line data from CAM2 in the second half of this year. Our ongoing phase 2 trial of oral KORSUVA is enrolling pre-dialysis stage 3 to 5 CKD patients with moderate to severe pruritus, with top-line data expected here in the second half of 2019. We also plan to initiate a phase 2 trial of oral KORSUVA in patients with chronic liver disease-associated pruritus, or CKD-aP, in the second quarter, following the recent completion of our phase 1 pharmacokinetic and safety trial. And then lastly, we anticipate initiating a multi-dose proof-of-concept phase 2 trial of oral KORSUVA in atopic dermatitis patients with moderate to severe pruritus around the middle of this year. So overall, we're well positioned to execute on our lead pivotal phase 3 program for CKD-aP and HD patients, as well as progress our oral KORSUVA program across multiple patient populations in 2019. So on the call today, I'll provide more detail on each of these programs, but I'd like to first briefly remind you of KORSUVA's profile, which we believe underlies its potential to act as a broad antipruritic agent across clinical conditions. KORSUVA is, of course, our novel first-in-class selective peripherally acting kappa opioid receptor agonist designed to function without traditional opioid side effects. due to its highly specific pharmacological action on kappa receptors and its chemical structure. KORSUVA's mechanism of action is mediated by kappa receptors on peripheral sensory afferents as well as on certain immune cells. The action of KORSUVA on dermal and epidermal immune cells blocks the release of a range of nerve-sensitizing molecules, or pruritogens, diminishing the stimulation of dermal sensory fibers. And we believe that this dual neuronal and anti-inflammatory effect affords KORSUVA an effective antipruritic action regardless of the initiating pathophysiology, whether that's chronic kidney disease, chronic liver disease, or some sort of dermatological dysfunction. So I'd like to start with updates on our lead program of KORSUVA injection in hemodialysis patients with CKD-aP. As a brief reminder, the late-stage clinical program for KORSUVA injection in this patient population currently includes 3 ongoing phase 3 studies: the CAM1 study, a U.S. efficacy trial, the CAM2 trial, a global efficacy trial, and an open-label 52-week extension safety study. We began enrolling patients in both CAM1 and CAM2 in 2018, and we recently announced both the completion of an interim statistical power analysis and the achievement of full enrollment of CAM1. Based on the recommendation of the Independent Data Monitoring Committee, or IDMC, we did not adjust the sample size of this trial. The IDMC's recommendation was based on its review of the results of a pre-specified interim conditional power assessment conducted after approximately 50% of the targeted patient numbers completed the 12-week treatment period. And to further clarify on this analysis, given the pre-specified decision rule and the formula used for calculating the conditional power, this outcome indicates that the results observed at the IA are consistent with the assumptions made regarding the sample size to maintain desired power to detect a difference between groups when we first planned the phase 3 trial. Following the IDMC recommendation, we completed enrollment of approximately 350 patients in CAM1, and we're on track to read out top-line data from this trial in the second quarter. Our second pivotal trial, CAM2, is also designed with a pre-specified interim assessment, again after approximately 50% of patients complete the treatment period. And similarly to CAM1, we look forward to providing the result of this analysis ahead of top-line data, which is expected in the second half of 2019. Both CAM1 and CAM2 are designed to investigate the efficacy of KORSUVA injection at a dose of 0.5 micrograms per kilo versus placebo. You'll recall administered 3 times per week after scheduled dialysis sessions over a 12-week treatment period. The primary efficacy endpoint is the proportion of patients achieving at least a 3-point improvement from baseline at week 12 with respect to the weekly mean of the daily worst itching intensity score as measured on a standard numeric rating scale, or NRS. Secondary endpoints in the trials measure aspects of itch-related quality of life using validated self-assessment scales, the Skindex-10 and the 5D Itch, which we employed in our completed phase 2B trial. Additional secondary endpoints include the proportion of patients achieving a greater than 4-point improvement from baseline in the weekly mean of the daily 24-hour worst itching scores at week 12. The third ongoing phase 3 trial in our pivotal program is an open-label 52-week extension study designed to evaluate the safety of KORSUVA injection in up to 240 patients. We now have close to full enrollment in this study, with about 125 patients through 6 months of treatment, and approximately 40% of these patients past the 1-year exposure level. To date, the safety and tolerability have been consistent with data reported in phase 2 trials of KORSUVA injection in hemodialysis patients, and based on a recently completed Independent Data Safety Monitoring Board evaluation, no new safety signals have been observed in this study. With top-line data from both CAM1 and CAM2 expected this year, We remain on track toward our goal of developing KORSUVA Injection as a breakthrough, first-in-class therapeutic for hemodialysis patients suffering from moderate to severe pruritus, for which there are currently no FDA-approved treatments. We also remain focused on advancing oral KORSUVA for pre-dialysis patients with moderate to severe CKD-aP. Based on generic pruritus-related script numbers, it is estimated there are approximately 2.5 million stage 3 to 5 CKD patients suffering from pruritus in the U.S., with current standard of care being predominantly generic corticosteroids and antihistamines. We're currently evaluating oral KORSUVA in an ongoing U.S. phase 2 trial of non-dialysis CKD-aP patients. This trial is a multi-center, randomized, double-blind, placebo-controlled, 12-week trial designed to evaluate the safety and efficacy of 3 doses of oral KORSUVA, a 0.25 mg tablet, 0.5 mg tablet, and a 1 milligram tablet administered once daily. From our phase 1 trial of oral KORSUVA, we determined that these tablet strengths provided exposure levels in CKD patients with moderate to severe renal impairment, which were approximately equivalent to those achieved with the 0.5 microgram per kilo dose of KORSUVA Injection in HD patients, a dose level which achieves statistically significant effects in both primary and secondary endpoints in our completed phase 2b trial. In this oral KORSUVA phase 2 trial, we expected to enroll 240 patients with 60 per arm. There's an unblinded interim power assessment at 50% enrollment for those who've completed the 12 weeks of treatment that allows the expansion of the study up to 480 patients. This trial is currently on track for patient enrollment, and we look forward to providing you with an update on the interim assessment in the coming months, and ultimately providing top-line data in the second half of this year. Lastly, as we move forward with additional pruritic patient populations that may benefit from oral KORSUVA, we're presently planning to initiate phase 2 trials in both chronic liver disease-associated pruritus and pruritus associated with atopic dermatitis. We've recently completed a phase 1 trial of oral KORSUVA at multiple tablet strengths in patients with chronic liver disease. The pharmacokinetic parameters were dose proportional, and oral KORSUVA was generally well tolerated with no unexpected safety signals reported. Based on this data, we expect to initiate a phase 2 trial in the second quarter of this year. We also expect to initiate a phase 2 proof of concept trial of oral KORSUVA in atopic dermatitis patients around the middle of this year, and we look forward to updating you on the details of this trial when we have it underway. So overall, we have a very significant year ahead of us here, with multiple late-stage trials expected to read out and an ongoing expanded development pipeline for additional pruritic populations. And we look forward to providing progress updates across all of these programs in the coming months. And with that, I'll now turn the call over to Manny, who will discuss our financial results. Manny.
Thank you, Derek. As a reminder, the full financial results for the fourth quarter and full year 2018 can be found in our press release issued today after the market closed. For the full year ended December 31st, 2018, we reported a net loss of $74 million, or $2.06 per basic and diluted share, compared to a net loss of $58.1 million, or $1.86 per basic and diluted share for 2017. For the fourth quarter of 2018, we reported a net loss of $20.7 million, or 52 cents per basic and diluted share, compared to a net loss of $14.2 million, or 43 cents per basic and diluted share for the same quarter of 2017. Revenues for the year ended December 31st, 2018 were $13.5 million, as compared to $911,000 in 2017. Revenues in 2018 were primarily related to our license agreement with Vifor Fresenius versus revenues of $843,000 in 2017 related to a sublicense fee received from a partner, Maruishi Pharmaceuticals, in connection with their sublicense agreement with Kasai Pharma. Revenues in 2018 also included $33,000 from sale of clinical compound to Maruishi, as compared to $68,000 in clinical compound revenue in 2017. For the fourth quarter of 2018, we recognized revenue of $5.5 million related to the Vifor Fresenius collaboration agreement. We did not recognize any revenues during the fourth quarter of 2017. Research and development expenses were $75.5 million for the year ended December 31st, 2018, as compared to $48.5 million in 2017. The higher R&D expenses in 2018 were primarily due to the increase in clinical trial costs, as well as increase in stock compensation and payroll-related expenses. For the fourth quarter, we reported R&D expense of $22.8 million as compared to $11.6 million in the same period of 2017, again due to increased clinical trial and personnel-related costs. General and administrative expenses were $15.3 million in 2018 compared to $11.9 million in 2017. The higher G&A expenses in 2018 were mainly due to increase in stock compensation and payroll and consultant-related expenses, partially offset by decreased rent and related costs. G&A expenses were $4.7 million during the fourth quarter of 2018 compared to $3 million in the same period of 2017. Other income was $3 million for the full year 2018 compared to $1.2 million for 2017. The increase in 2018 was primarily due to higher balance of investments in this period. Other income was $1.2 million in the fourth quarter of 2018 compared to $368,000 in the same period of prior year. As of December 31st, 2018, our cash equivalents totaled $182.8 million compared to $92.6 million at the end of 2017. The increase primarily resulted from net proceeds of $92.1 million from the company's follow-on offering in July, proceeds of $70 million related to the license agreement with Vifor Fresenius, which included an upfront payment of $50 million in cash and $20 million of equity investment at premium, and the proceeds from exercise of stock options. Turning to our financial guidance, based on the projected costs of our clinical development plans and timing expectations, we expect that our current cash equivalents as of December 31st, 2018 will be sufficient to fund our operating expenses and capital expenditure requirements into 2021 without taking into account any potential milestones under existing collaborations. I'll now turn back the call to the operator for Q&A.
Ladies and gentlemen, at this time, if you have a question, please press the star then the number 1 key on your touch-tone telephone. If your question has been answered or you wish to remove yourself from the queue, please press the pound key. To prevent any background noise, we ask that you please place your line on mute once your question has been stated. And our first question is from Chris Howerton from Jefferies. Your line is now open.
Great. Well, congrats on the quarter and all the progress, and thanks for taking the question. I think, Derek, the main question that I have is maybe just helping contextualize the interim analysis, the powering analysis that was performed earlier this year. I know that you kind of described the process, but it might be helpful, at least from my side, to better understand what the other potential outcomes would have been. So let's say that they did say you needed to increase the sample size. What would have triggered that? And maybe you could help us understand what the alternative outcomes could have been.
Yep, sure. Hi, Chris. Thanks for that. Yeah, so this was really set up in a standard way for this type of analysis. So what we asked the IDMC to calculate is the required sample size to maintain a desired statistical power to see a difference between the two groups in the study. And their possible advice in relation to that was graded by sample size. So there was a possibility to increase the sample size to X, which was less than maximum, to maintain that power, or to increase it to Y, which was full sample size in the study, which was up to 500. Or there was a possibility to not increase the sample size as the desired power to maintain a difference between the two groups was evidently maintained. And remember, of course, the IDMC runs this analysis in an unblinded fashion. And that was the specific advice we received for the IDMC. And what that tells us is that our assumptions, again, made based on our odds ratio or effect size observed in phase 2B, were correct in terms of the required sample size to maintain a high level of power to see a difference between the two groups. So really what they're modeling in their analysis is maintaining a threshold of statistical power to maintain a difference.
Okay. So essentially they observed a sufficient effect or powering and able to meet the initial powering assumptions that you had when you designed the trial. Is
correct.
The basic story?
Correct.
Yeah. Okay.
And obviously we'd set that threshold at a high level, a conservative level.
Right. Okay. And then for the chronic liver disease phase 1 trial, I know that you kind of described the pharmacokinetics and the safety. There was nothing unexpected. Are there any plans to present those data in a more full way?
There is, and we're actually going to present some summary PK data when we initiate that phase 2 trial. You're going to see some overall exposure data, again in a similar fashion to the chronic kidney disease phase 1, where we wanted to essentially maintain an overall exposure that would match the efficacious dose we've used in the hemodialysis patient population. So you're going to see that data in summary fashion when we initiate the phase 2.
Okay, that makes sense. And then maybe just one last question, if I may. For the atopic dermatitis trial, I think you mentioned that we'll see some more information about that when the trial is actually up and going. But can you give us any preview in terms of what patient population you're planning to target with that?
Yeah, we're going to look at a breadth of patient population, both in terms of pathophysiology. We'll be looking at both the mild to moderate and the moderate to severe. And obviously there will be inclusion criteria related to moderate to severe pruritus within those populations.
Okay, all right. Well, congrats again, and thanks for taking the questions, and I'll hop back in the queue. Appreciate it.
Thanks, Chris.
Thank you. Our next question is from Jason Gerberry from Bank of America. Your line is now open.
Oh, hey, good evening. Thanks for taking my questions. I guess just first question, just on the post-operative applications for 845. Have you had any recent FDA interactions? It sounds like maybe there weren't. I wasn't sure exactly. I know there was potentially some discussions just regarding kind of framing the pathway for the PONV indication. And then secondly, can you just talk a little bit about, when we think about the phase 2 trial for oral KORSUVA in the CKD-3/5 group, can you just talk about what you think about as more of a clinically meaningful effect size in that group?
Yeah, thanks, Jason. To the first of those on PONV, our aim there is to get clarity from the agency, both on the regulatory path there and on safety exposures and how already existing post-op exposures would fit into that particular program if we change the label on that. And we will certainly guide when we initiate the next possible PONV trial as to some more detail in relation to regulatory path there. And that's something we'll provide. On the CKD oral, we don't pre-specify clinically meaningful differences. This is really related to statistical significance. There we will be looking at differences from baseline and measuring that on the usual NRS scales in terms of mean changes and worst itching scores. But we don't set those up with pre-specified differences. We simply model those in terms of sample sizes, if that's what you're getting at, based on effect sizes we've already observed with KORSUVA in established populations. And obviously the main effect size we model that on is related to CKD-aP and hemodialysis patients.
Got it. Thank you.
Thanks, Jason.
Thank you. Our next question is from David Amsellem from Piper Jaffray. Your line is now open.
Thanks. So I had a couple questions about the atopic derm study that you're planning to initiate later this year. So is that going to have a primary outcome measure looking at a 4-point responder analysis on worst itch, or the 3-point responder analysis? And just help us understand what your expectations are, given that, at least in dermatologic conditions for other antipruritics, the 4-point responder analysis seems to be something that the FDA wants. And then secondly, on the atopic derm program, any color on dosing and how that may or may not differ from the other doses you're evaluating in the CKD and CLD programs? Thanks.
Yeah. Thanks, David. No, and that's a great question, and as you know, we've discussed this before in terms of the appropriate endpoint for each of these patient populations. So, the endpoint we reached for the hemodialysis-associated pruritus population was really based on our empirical analysis in relation to our breakthrough application, and that was something we looked at in collaboration with the FDA. So that was taking data from patients where we see significant differences in their worst itch scores and correlating that with their quality of life scores and the patient impression of change. And we empirically derived what would be a clinically meaningful change for those particular patients. And from that, we derived a number somewhere around 2.5 on an NRS scale would be clinically meaningful, and we bumped that to 3, and that's what we ended at in the phase 3 trial that's underway. You are correct. There has been a dogma within the dermatological populations for a 4-point reduction, really based on an analysis that was only performed within the psoriatic population, looking at clinical meaningfulness, but nevertheless a dogma. So our plan in moving oral KORSUVA into the AD population is indeed to look at that level of reduction as well as mean change from baseline. So we will be looking at the 4-point when we move to the dermatological population, and we're still continuing with 3-point, which we've again derived empirically in discussion with the FDA for the CKD-associated population.
And then dosing, any color on dosing for the AD study?
Yeah, we'll guide to that when we initiate the trial. Of course, that's likely to be twice-a-day dosing, because unlike with the CKD population, we have a very healthy, generally population with AD, healthy renal function. So that's likely to be a BID dosing regimen in that population, and we'll guide to the dose when we start the trial there.
Okay. Thanks, Derek.
Thanks, David.
Thank you. Our next question is from Charles Duncan from Cantor Fitzgerald. Your line is now open.
Hi, guys. Thanks for taking my question, and congrats on a good year of progress. Looks like a pretty interesting one coming up. Couple of quick questions. You laid out very nicely the interim analysis kind of criteria in CALM-1. Can you tell us, is CALM-2 exactly the same or different? And also, when you look on a blinded basis at the patients being enrolled in CALM-2, how does that compare to CALM-1?
Yes, I can't give you the answer to the last one, Charles, because no one lets me near any sort of data on a blinded basis to look at, so we don't look at that routinely. But in terms of the design of the pre-specified interim assessment, exactly the same setup as we have for CAM1. We're going to look at that when 50% of the patients complete the 12-week treatment period, and as I just talked about with Chris, it's going to be the same conditional power analysis. Again, we remain blinded, the IDMC is unblinded, and they look to maintain a conservative level of power, which is the same as CAM1, to see a statistical difference between the two groups in that trial. So it's designed the same way. It's going to be performed at 50% of the patients completing the 12-week treatment period.
Okay, that's helpful. So they don't let you near data on an unblinded or a blinded basis. How about on unblinded?
Yeah, the end of the trial I get to see it. That's about it, Charles. So it's a horrible life up here.
Okay, great. So also on CALM, I'll call it CALM-3, the 52-week extension study. I don't know what you call it.
Yeah.
You mentioned a certain number of patients have gotten through 6 months and then 12 months. And I guess that means that they continue on the regimen of getting the drug infused in dialysis, and how are they doing in terms of concomitant meds or any other call it qualitative look at how those patients are doing over time?
Yeah. No, so we don't. There's no other measurements in that particular trial, Charles, other than safety. But you're correct; those patients are dosed in exactly the same manner as we use for our phase 3 trials, and in exactly the same manner that this drug is going to be used commercially. So that's 3 times a week post-dialysis sessions. And I know, just to remind you, because the drug's renally excreted, we get this depot exposure level for the next 2 days post-dialysis, which maintains the antipruritic activity. So they're dosed in exactly the same manner 3 times a week for up to 1 year. We have, what I'd said is we have approximately 125 patients that are through the 6-month mark, and about 40% of those are up and through the 1-year mark. And to date, we've had regular independent data safety monitoring board analysis on that trial. We just completed one this quarter, and there was no unexpected safety signals within that group at all. Okay,
that's helpful. And then just a couple of other quick ones, because it's been a couple of years since I've looked into this. The IP on KORSUVA lasts, the new chemical entity IP lasts through when? And then can you help us understand whether or not there's call it a picket fence around other non-KORSUVA molecules that may, extend IP?
Yeah, so there's no, so there's about a dozen issued patents, U.S. patents, I presume you're asking about. Well, obviously, we've filed worldwide, but there's a dozen issued U.S. patents, including composition of matter, of course, and that patent runs to 2027 without extension. So with the 5 years, that goes to 2032. Beyond that, our patent strategy is the normal one. We have obviously use patents around where we're using the molecule. And then beyond that, we also have some formulation-related patents. And those run into the mid-2030s, in fact, that we've been working on. So there is a number of, if you like, fences around the sequences. In terms of, likelihood of anyone breaking those patents, unlike small organics where that's much more possible in terms of manipulating the chemistry around these heterocyclic rings, with peptide sequences much more difficult to break, Charles. So we've actually, we think, secured the entire area, and as you know, we tested this empirically a number of years ago on tetrapeptide sequences that have high affinity specifically for the kappa receptor, and of course that's our primary claim in relation to most of those patents. So very difficult to break a sequence patent, and so we think we have that quite well controlled in terms of the entire tetrapeptide sequences that can interact specifically with kappa opioid receptors.
And last question related to going back to David's question on atopic derm. I'm wondering if you have a perspective on, kappa opioid agonist mechanism versus NK1 antagonist mechanism. There's a fair amount of activity in terms of pruritus associated with atopic derm with the NK1 antagonist. I know that you've placed your bet, but love to hear your perspective on the relative merits of the two mechanisms.
Yeah, well, it's a big opportunity. I think there's room for everybody, and there's certainly a significant unmet need in that patient population. our view is that, and I think the physiology agrees with this, that the endogenous opioid system locally is a very important control mechanism for pruritus. And, one of the animal models we've used and the industry uses injection of a kappa antagonist to produce a fairly severe pruritus in animals, which is an indication of how important that system is. And my view, and as you know, I've done this for a couple of years now in GPCR drug development, is that agonism is by far the most potent mechanism when it comes to human therapeutics. And so I think we have a driving system that we're amplifying, which is always nice, and it's much more difficult to antagonize, especially in that situation. So we think we're tapped into one of the main driving systems, and so far that's proven to be correct, as we're seeing efficacy across all animal models we look at with the drug, regardless of the mechanism. And we know from our data and some other Japanese clinical data that this mechanism is certainly effective both in chronic kidney disease and chronic liver disease associated pruritus. So far the data would bear out the broad antipruritic applicability, but we're going to test that empirically. So stay tuned for about 12 months, and we're going to have the answer to all of this.
Okay, very good. Helpful reminder of Pharmacology 101 with the agonist statements. Thanks for taking my questions.
Thanks, Charles.
Thank you. Our next question is from Annabelle Samimi from Stifel. Your line is now open.
Hi. Thanks for taking my question. Just wanted to go back to the CLD population. Are there any particular adverse events, side effects that you might have to watch out for in this particular population, given the complexity of their disease, any problems? And then with regard to the dose proportionality, should we expect something similar to what you're already looking at in CKD? And do you have any sense of the size of the trial? Thanks.
Hi, Annabelle. Thank you. Yeah, so one of the advantages of this chemical class, of course, you remember, is we don't have any liver metabolism, and we don't have any interaction, having performed every possible test on this with any of the CYP enzymes, either as a substrate or stimulator or inhibitor. So we didn't predict we'd have specific issues with this patient population, but as you rightly point out, there's a lot of comorbidities in there. So we wanted to empirically ensure that the exposure levels we could attain there were going to match what we knew was efficacious based on our CKD data. And we've done that, and we're going to select tablet strengths that are going to give us that. Difference between this and the CKD oral is going to be, as I said earlier, we're going to be dosing this twice a day. Despite the fact that some liver patients do have some inhibition of renal function, it's not to the level that we saw significant diminishment in excretion. And as you know, 845 or KORSUVA is excreted almost exclusively via the urine, and so we wanted to establish that and make sure that was the case. So safety looks good. We don't see anything different there than we've seen in other populations, and when we start that trial, we'll announce the tablet strengths that we're going to use in that particular population, which is going to be very similar in range to the dosages we're using in the CKD stage 3 to 5 patients. The difference, again, being there we can dose once a day. This is likely to be a twice-a-day dosaging.
Okay. And just on the opportunity in CLD, so you mentioned CKD is about 2.5 that has stage 3 to 5 chronic kidney disease. Is CLD similarly sized? Is it much larger? Is it smaller? Just can you give us a sense there?
Yeah, no, that's also a good question. Again, we looked at that in a similar fashion, looking at script numbers rather than epidemiological numbers, and using ICD-9 codes to cross that with the various disease states and liver. Various liver conditions are associated with moderate to severe pruritus. Of course, primary biliary cholangitis being the best known, but viral infections, Hep B and Hep C, some NASH patients, some cirrhosis patients. When you add all those up and look at that population in the U.S., and of course, as we know from recent data, some of the some of the medications associated with liver disease situations can also induce pretty severe pruritus. And when we add all of those up, it looks like somewhere around a couple of million scripts a year when we look at all of those conditions. So that's a significant opportunity there, which is somewhat similar to the pre-dialysis CKD opportunity. And then, of course, when you get to atopic, then that's a different magnitude there in terms of potential addressable patient population.
Okay, great. And then while we're on the atopic dermatitis topic, are there any, I mean, it's been a difficult, elusive disease to treat from a pruritic condition. Is there, or from a pruritic perspective, are there any teachings or anything that you can learn from other trial shortcomings, trial failures that you can apply to your program that might help with a better outcome?
Yeah, no, I think, in reality, Annabelle, most of the pruritus models that we use and the industry uses are dermatological of one sort or another, and we certainly had KORSUVA through all of those and shown great effect. Certainly in contrast to some other mechanisms in those models. But at the end of the day, there really hasn't been a selective peripherally acting kappa tested in that population. Everything that's been used to date has been less selective or partial in terms of agonism. So we feel as though there hasn't been an appropriate test yet in that patient population, and we'll be careful how we select patients coming into that trial. So mechanistically, that's something I think we have to look at empirically, but based on all the data we've generated both preclinically and the fact that it seems to work, that is this mechanism, peripheral kappa, across other patient populations, we're optimistic that we can have a beneficial effect within that patient population.
Okay, great. Thank you.
Thanks, Annabelle.
Thank you. As a reminder, ladies and gentlemen, if you have a question, please press the star, then the number 1 key on your touch-tone telephone. And our next question is from Arlinda Lee from Canaccord. Your line is now open.
Hi, guys. Thanks for taking my questions. Can you remind us on what the exposure requirements are for filing and what you will have by that time? You mentioned some statistics on the open and label extension, and I'm curious how many more patients or duration you'll need to have by the time you file. And then also, could you provide some, you've talked about getting additional regulatory clarity on the postoperative pain, and I'm wondering how high of a priority is that for you guys right now? Thanks.
Yeah. So on the last question, thanks for the question. On the last one, as you know, as we've said pretty consistently, our main priority here is pruritus, and that's where we're focusing our effort. The PONV effort is really to get clarity in relation to the endpoints required there for labeling, regulatory path, and then some clarity on some of the work we've done so far there in terms of safety exposures. And with that package, I think we'd be in a much stronger position to look for an appropriate partner to come in and help fund any effort we have on the PNV side. So that's the strategy in relation to that application. For the first question, ICH guidelines on exposures for the KORSUVA injection program are 1,500, and that's how we've designed our program. So both the phase 3s that are ongoing have safety exposure extensions beyond those trials, and as you know, we started the 52-week extension study that I mentioned on the call in 2017. So we've been, making sure we can get to those exposure levels and looking ways to add up exposures to get the fastest possible path to NDA. As you know, it's a breakthrough designated compound. There is a possibility that we can file with less safety exposures with such a compound. It's certainly something we will explore with the FDA. We're not counting on that path, but once we have our first U.S.-based CAM1 pivotal data, it's certainly something we'll explore with the FDA, but we've designed our whole program to accommodate normal ICH guidelines, which is 1,500 exposures.
Okay, great. Thanks.
Thanks.
Thank you. Our next question is from Alan Carr from Needham. Your line is now open.
Hi, thanks for taking my questions. Just to follow up on the last one, is it feasible for you all to file NDA, MAA by the end of this year after completing both COM1 and COM2? And then can you also comment on what non-clinical work or items are needed for the NDA? Where do those stand? Thanks.
All right, Alan, let me take the last one first. Non-clinical work, we're basically complete. Almost everything we need in the non-clinical sense, preclinical sense, is finished. And so what we're working on now is, as you point out, completion of, excuse us, we got a fire occurring somewhere nearby. We're looking to complete our 2 pivotal CAM1 and CAM2 studies, and then our safety extension studies on both of those, of course, and then file as quickly as we can afterwards. So we'll be able to guide more fully, more accurately, if you like, on this a little later this year, once we have our readouts from CAM1 and we know where we are with CAM2, and we've advanced our 52-week safety study. This is something we can guide to a little later this year.
What sort of meeting would that be with the FDA after your first phase 3? Would that be a formal end of phase 3 or something else? How would that work?
Yeah, so fortunately with a breakthrough designation, we can request meetings fairly frequently. So we probably reserve our pre-NDA meeting for a little later, and we'll use another, standard meeting to interact with the FDA. Okay, and then last one, any updates on any of your collaborations with in Japan or Korea in terms of how they're progressing in development? Both are progressing. The CKD, or Chungkondang Pharma, is working with us in relation to Korean participation in the global phase 3. Maruishi, as you know, has sub-licensed commercial rights in hemodialysis patients to Kisai, and they're advancing that program well, and we'll certainly report outcomes in both of those areas when we see the data. Great. Thanks for taking my questions. Thanks, Alan.
Thank you. Our next question is from Esther Hong from Janney. Your line is now open.
Hi, thanks for taking my question. So my first one is on partnerships over IV KORSUVA in hemodialysis patients. So in the U.S., you've got a significant partnership with Vifor Fresenius, one of the largest dialysis center owners in the U.S. So have you had discussions with the other largest owner, DaVita? Do the terms of your Vifor Fresenius agreement allow you to also partner with DaVita? And then beyond that, how would IV KORSUVA roll out commercially in the Fresenius clinics? Would they be specific to certain regions? Could we imagine that they would be in the majority of clinics? How would it look? Thanks.
Yeah. Thanks, Esther. To the last point, so the deal we have, the license agreement we have with Vifor Fresenius is really focused on ex-US. So they have European rights and some territories in the Asia-Pacific region, obviously not Japan and South Korea. Within the US, we have a co-promotion agreement specifically in Fresenius clinics, and that's about 38% of the patient population in the US. So the vast majority, I guess 62%, remains solely in Cara's purview, and we are free to market within that population solely, and we will do so and are planning to do so. So that's the difference in setup. What was your other question in relation to Fresenius? Oh, so the Fresenius arrangement in no way restricts us on how we can interact with other dialysis providers in the U.S. We can set up any type of co-promotion licensing agreements with any of these dialysis providers.
Do you expect to have discussions with DaVita, or is that an option?
We do. As we advance our commercial plans, we will be not only DaVita, but some of the other major independent dialysis providers we'll be talking to.
Okay, great. Thank you.
Thanks,
Thank
Esther.
you. Thank you. At this time, I am showing no further questions. I would like to turn the call back over to Derek Chalmers for closing remarks.
Great, thank you. And thank you, everybody, for participating in today's call. I'd like to thank the Cara team for their continued hard work and commitment to supporting our various programs, our study investigators, and most importantly, all of the patients who continue to participate in our clinical trials. So thank you, everybody, and we look forward to updating you very, very soon. Thank you very much.
Ladies and gentlemen, this concludes today's call. Thank you again for your participation. You may now disconnect. Have a great day.